thank you for the kind introduction and welcome everyone. As already said, today I will present a work which was already published in 2016 in TVCG, and on behalf of my colleagues from TU Wien and Masaryk University, I will tell you something about this paper which focuses on the intuitive exploration of a protein tunnel along its center line and its surroundings over time. So I will start first with a short introduction into the world of proteins. The proteins are highly complex macromolecules, and their understanding of their biochemical functions and structures can help us to reveal the fundamentals of the biochemical processes taking place in each living cell. However, this process of finding the understanding or getting the insight into the proteins is highly time-consuming and demanding on the resources. This is mainly due to the protein dynamics, which changes the protein shape and its biochemical properties over time. At our university, I cooperate with the domain experts which are interested in the protein engineering. This basically means that they are looking for a proper combination of a protein and another small molecule called ligand. They are looking for a combination of these two which leads to the desired reaction. But before such reaction can occur, the ligand actually has to reach an active site, by which in this presentation we understand the deeply buried site which is surrounded by the active amino acids which are taking part in this particular reaction. The active site is interconnected with the protein surface by a empty void which we called a protein tunnel. And these protein tunnels basically serves as a potential transport pathways for these ligands. Whether the ligand will pass through the tunnel depends on many factors. One of those is the size of the protein tunnel. There are parts of the tunnel where the ligand fits very well. However, this doesn't have to hold for the whole tunnel, and there might be places which are too narrow for a ligand to pass through, and we call these the tunnel bottlenecks. Currently, all the methods which are focusing on visualizing the protein tunnels and its surroundings over time are doing those mostly in 3D using tools such as PyMol. But there is a problem which has two phases. First, as the phenomenon is visualized in 3D, some common problems such as occlusion occurs. And second, currently we are able to generate the long molecular dynamic simulations with hundreds of thousands of time steps. And it's really hard or nearly impossible to comprehend all the details from the 3D visualization of this problem. So we ask ourselves actually a question, what do biochemists want to see in their data? And we found out that they are more interested in the statistical overview of their protein than seeing the actual visualization of the simulation itself. So we generated new representation, or which we call AnimoAminoMiner. And basically this focuses on an aggregated view of a protein tunnel and its surroundings over time in a single image. It's actually multiple view visualization. The upper part we call Tun Profile, and the bottom part is called Amino AcidExplorer. The Tun Profile focuses on the visualization of tunnel length and width, and their changes over time. The width here is computed as the radius of the tunnel sphere in the tunnel representation and is depicted on the y-axis, while the tunnel length is depicted on the x-axis, and the part closer to the active site is visualized on the left, and the part of the tunnel closer to the to the surface is visualized on the right. The tunnel shape and its corresponding curve is visualized and computed in each time step, and this way we gradually build a single aggregated overview which immediately gives us a representation of the tunnel stability over time. The stability is now here visualized as a dense packing of curves, and using this representation, the biochemist can actually also see the positions of the individual bottlenecks along the tunnel center line. The tunnel shape and its biochemical properties highly depends on the tunnel surroundings. And here comes into play our bottom part of the visualization. In order to map the 3D position of each individual amino acid to 2D, we need to use some abstraction. Here we emphasize the fact that the actual position of individual amino acids around the tunnel is not really crucial for the biochemical properties of the tunnel itself. We rather emphasize the fact that some amino acids which are closer to the tunnel or influence the bigger part of it are more important, and therefore we depict them by default on top of the visualization. And the amino acids farther away, we depict lower. Whether an amino acid influences a tunnel and which part of it is computed as follows. First, we find a closest sphere for each atom of the amino acid. This way we obtain a separate intervals of spheres. As a second step, we evaluate the distances between these intervals, and if the distance is lower than 3 angstroms, we mark the spheres in between also as influenced by the given amino acid. And we continue in the similar manner. The threshold 3 angstrom is not selected randomly. This comes from the fact that our domain experts consider the 3 angstrom to be a threshold for a single amino acid to be influencing the tunnel in the first place. Once we have merged all the intervals, we can now render a bar into our chart visualization and repeat the process for another time step. And do it again and again until we get the whole visualization for a single amino acid over time. Here each line is sorted according to the time from top to bottom, and then we can repeat the process again for each amino acid in the data set. The individual amino acids can be sorted by the user-defined ranking, which can be either the biochemical property or the importance of the single amino acids. I will get to this a little bit later. If the biochemists would consider the first view on the top to scattered, we also provide them with the blended view, which shows more important features of the temporal contribution of the amino acids. So the Amino AcidExplorer, together with the Tun Profile, provides a complete overview of the tunnel over time. But the main strength of this tool comes from the possibility to interactively explore both its parts. The top part, for instance, can be colored by various properties, such as time in this picture, to communicate important features of the simulation itself. Similarly, the bottom part can be colored by various biochemical properties, such as hydrophobicity, partial charge, or the importance of the individual amino acids. The biochemist can involve or explore the contribution of each amino acid with respect to their distance from the active site using a vertical slider. The biochemist can involve or employ the horizontal and vertical zooming and panning in order to explore individual amino acids in more details. And moreover, if they click to a selected amino acids, a table view with various information pops up. In this view, the biochemist can see several things. On the top, they can see the part of the amino acid chain or the sequence where the selected amino acid belongs to. The middle part shows the actual values of individual biochemical properties. However, the most important part is the bottom one. Here, the biochemist can observe the influence of this selected amino acids to another tunnels, not that one which was already shown, but to another tunnels which are presented in the molecule. And this is highly important, especially when they are trying to mutate the amino acids to replace it by some other amino acids in order to change the properties of the protein. They can immediately see which structures of the protein will be influenced and changed as well, and whether this is desired or not. If the biochemist would like to focus on the smaller time span or some particular feature in the data, they can employ various selections. For instance, here is a vertical selections on the curve, and the selected or all the remaining curves can be filtered out. A similar filtering can be also applied to the amino acids itself. The biochemist can currently employ, sorry for the quality of the image. The biochemist can basically apply 4 different thresholds for 4 different biochemical properties in the data set. And the resulting filter is actually a combination of these criteria. The biochemist can easily also invert the thresholds in order to filter out the amino acids below or above the selected value. The same criteria can also be used to sorting the amino acids. So you can see that amino acids can be sorted by the hydrophobicity. So you can see them split green and violet, or they can be sorted by the importance of the influence. So one more, here it's on the top. Finally, our tool can be also linked with another representation which already exists out there. Here you can see the view. In the right view is the representation which represents the tunnel using the contours. So the biochemist can use the vertical slider to pick up the particle location along the tunnel center line, and the corresponding actual shape and actual positions of individual amino acids around this position can be computed. The view is also interactively linked with the 3D view. So here, for instance, you can see that the biochemist can click on individual amino acids, and these amino acids are highlighted in the 3D, and they can then explore the spatial orientation with respect to the actual tunnel. The method was tested and evaluated by domain experts from Protein and Metabolic Engineering Laboratory at the Masaryk University. And today I will show you one of the case studies we did. We actually used existing case study, which goal was to increase the protein reactivity with the given ligand. We used a protein from the haloalkane dehydrogenases family, and the main problem here, as you can see, is that the tunnel bottleneck is really scattered over time, which basically gives no chance for a ligand to pass through. So the biochemist needed to replace some amino acids with the smaller ones in order to change the protein. So they used our ranking system, and by this they were able to pinpoint the most important amino acids which influence the given part of the tunnel the most. So they replaced this phenylalanine by a significantly smaller amino acid glycine. And as you can see, now the glycine is lower in the ranking system because it doesn't influence the tunnel that much since it's smaller. And the most importantly, the protein bottleneck is completely gone. So now the protein actually can really interact with the given ligand. One more side by side, you can see by changing a single amino acids, the biochemists were able to significantly change the protein function. So which brings me basically to the end of my talk. I've shown you a tool for visual exploration of tunnel along its center line, which works really well for smaller dynamics. It works also for the longer dynamics, but we are currently working on having a better representation or more sophisticated representation of the amino acid part than the naive averaging as shown in this visualization. Since I have some time, I would also do a little bit more advertisement. As you might know, this year BioVis will be taking place not far from here in Brno. And myself, together with Michael Krone, are considering to hold a workshop on the molecular visualization, which should be focused on the application-oriented papers. Currently, we are thinking about having mostly a WebGL representation and visualization for molecules, but if you would like to hear more, talk about it, or have some suggestions, you can always find me during coffee breaks. With that being said, thank you for your attention, and if you have any questions, feel free to ask.
Okay, thank you, Jan. Maybe there are questions? There's not too many people standing up, so I have one question. I was just wondering about how intuitive is this sort of representation for your biochemists that you collaborated with. So I think they are used to looking at this stuff in 3D, and now you have this very sort of abstract representation. So
yeah, actually, it is high. During our testing, we found out that it was quite interactive or intuitive for the biochemist, mainly because the ranking system. So if if you can see in this visualization, the biochemist basically can see really the importance of individual amino acids along the tunnel. So if you look at it in the 3D, you spend a lot of time rotating the 3D view to finding your amino acid, looking where it is, how it positions,
Right,
etc.
and you see this in one.
You can immediately here see where exactly it is with respect to your tunnel, whether it influenced the bottleneck or the bigger part of the tunnel, and how much it is influencing the tunnel over time, because as you can see, some amino acids down here will influence only small part of it,
Right.
or are involved in the interaction only for a small portion of the time. And then if you find out 2, 3 interesting amino acids, you can always click on them and see them in the 3D anyway.
Okay, clear. So if you have the time, I think I will move to the next speaker so we can catch up a little bit. So I would like to ask Henry to come forward.